I'm Senior Life Sciences Analyst John Vandermosten, and I'm joined by the CEO of TuHURA Biosciences, Dr. James Bianco. TuHURA is working on three main products, including IFX 2.0, an anti-VISTA asset, and its delta opioid receptor. Their most advanced program is their IFX 2.0, and we have Dr. Bianco with us today to tell us more about it. So, Dr. Bianco, you have these programs in place, and you started your trial for IFX 2.0 in the middle of last year, in 2025. Tell us how those have progressed so far and how enrollment is going.
Sure. Thanks for having me, John. Right now, the phase 3 study has almost all of the sites up and initiated and is enrolling patients. And then the phase 2 study that we've talked about in a minute is also enrolling patients and potentially have preliminary data at the June American Society of Clinical Oncology meetings this year.
Okay. And one of the interesting features of your IFX 3.0, your IFX 2.0 program and your phase 3 is that it has been designed to satisfy the requirements for both accelerated and full approval by the FDA. How did you work that out with the FDA, and also how does that save time and money for TuHURA as you advance the program in Merkel cell carcinoma?
Good question. You know, we worked with the division and then with a group called the Oncology Center of Excellence. In 2017, what used to be called the Office of Oncology Drug Development, name was changed to the OCE, the Oncology Center of Excellence. We worked with the deputy director who really proposed a kind of an interesting trial design, which we're really excited about. Probably the wave where you're going to see these types of studies, accelerated approval trials move in the future. The first one was this Project Frontrunner initiative. So they saw our data for IFX in patients progressing on Merck's Keytruda and saw those data as encouraging enough to ask us to move it into the frontline treatment setting, do a randomized trial, Keytruda plus IFX 2.0 versus Keytruda, and see if we can improve the response rate to that drug in the 50% of patients who don't respond with this terrible cutaneous skin cancer called Merkel cell carcinoma. So that was the first aspect, right? Moving from a second line setting potentially into the frontline setting. In that trial, they've also encouraged us to use a key secondary endpoint. So for accelerated approval, the endpoint comes relatively quickly. You'll find out if you have been successful within 6 months after the enrollment's completed, where that approval is based on what they call a surrogate endpoint likely to predict clinical benefit, which is response rate. So if you hit response rate, you can get accelerated approval. And that can happen within, like I said, total time from start of the study to approval can be as short as 24 months. But they then recommended that we incorporated a key secondary endpoint, and that's called progression-free survival, which is a clinically beneficial endpoint. So it's not a surrogate. And if we incorporate that into the trial design, then we don't have to do what is typically required, which is what's called a post-approval confirmatory trial. And those trials are typically very expensive, take a long period of time. And so we don't have to do that. So we have the ability for a single study to give us an answer within 24 months from start to finish and 18 months start to finish, and approval potentially in 24 months. And then about 14, 15 months later, when that trial matures, if we hit the PFS, progression-free survival endpoint, that study will flip from accelerated to full approval. And again, pushing it into the frontline setting, the addressable market size is considerably larger, right? And you're now talking about 3,000 patients, then potentially 1,100 or 1,200 patients.
Okay, I just wanted to reiterate the trial design there that really is a big time saver and money saver. You're going to be using the same patients for the accelerated approval as you will for the full approval, and you'll also save time because you won't have to start another study and wait for that that confirmatory endpoint to be met. Is that right, Jim?
Yeah, I mean, there's a good example right now. I mean, I'm an investor in Replimune for full disclosure. They have a drug that tries to work similar to what IFX does. They had a successful study in third-line melanoma for accelerated approval. So they did a single trial, response rate for the endpoint, they will receive accelerated approval on this resubmission, but they're required to have a post-approval confirmatory trial, which they have ongoing. That study has 400 patients, will be over approximately 48 months. And, in my experience, 400 patients over a 5-year study is probably $60-70 million. We don't have to do that. And in fact, we're not in the second line setting. We got moved into frontline, and we save that whole, quite frankly, the risk associated with that. And one feature that we didn't mention is we did this under what's called a special protocol assessment agreement. So it's a binding agreement with the FDA that if we meet the endpoints per the protocol and carry out the study per the protocol that the FDA agreed to, then we should get accelerated approval and full approval at the end of the process.
That's great. A great time saver. Now, you're also running a phase 2 study for IFX 2.0 in Merkel cell carcinoma. What's the difference between the population in the phase 2 compared with the phase 3?
That's a good question. So the primary issue with checkpoint inhibitors, which, as you know, has just revolutionized the treatment of cancer. I mean, it's a GLP-1 for oncology, if you will. Merck sells $30 billion a year for Keytruda, but only 20% of patients respond to it. So the majority of tumors don't look foreign to your immune system. So just like what we're doing in the phase 3 trial, where we're trying to make those patients tumors that don't trigger an immune response, we're going outside of Merkel cell, and we're looking at classically what they call non-immunogenic tumors. Ovarian cancer, pancreatic cancer is notoriously resistant to any checkpoint therapy. And so these are patients who have deep-seated tumors, typically in their abdomen or in their liver. We will administer IFX to those tumors and give them Keytruda and see whether or not we can get a response. And if we show that biologically we can do that, like we showed in Merkel cell, then that addressable market is commercially very attractive, right? Because now you're talking about the 80% of tumors that typically don't respond. So it's really more of a proof of concept that in terms of the generalizability of IFX 2.0 outside of just a relatively limited Merkel cell carcinoma population into much larger tumor types that classically will not respond to checkpoint inhibitors like Merck's Keytruda.
So if the phase 2 trial in deep-seated tumors is successful, what are the next steps for that for approval? And could you combine that with what you're doing in the other Merkel cell carcinoma trial, the phase 3?
So, where we would see if we are successful again, it won't take many patients, right? If you take 10 or 12 patients with tumors that they don't even recommend a checkpoint inhibitor for, because they have notoriously failed in clinical studies with checkpoint inhibitors, and you convert a good portion of those patients into having meaningful clinical responses, I think that's a discussion you have with the agency. Specifically, accelerated approval is certainly possible in that setting. And this, again, if you think of precision or targeted therapies, targeted medicines, the biologic reason why these tumors don't respond is no different if it's Merkel cell or pancreatic or ovarian. And so we call this a basket trial, where you're not focused on a specific tumor type, you're focused on a specific abnormality or biology in that tumor that it's allowing your drug to work when other drugs don't.
So I wanted to move on to what I think is probably the most interesting thing that you're working on, and it actually is represented by the poster that's behind you, which is the delta opioid receptor. Now, this is a fascinating area for a bunch of reasons, which we'll get into while we're talking about it. But this is really cutting edge stuff that you're working on, and you had a presentation at ASH just a couple months ago. And can you share with us what you found about the delta opioid receptor and its inhibition on immunosuppression, and what was in that poster?
Sure. I think probably it's good to just put it in a large context, right? People always talked about, when you have too much stress, your immune system is suppressed, and you can have an infection if you receive drugs like morphine for pain after surgery, et cetera. So we're the first to link that a specific opioid receptor called the delta opioid receptor is highly expressed on immune cells. And the type of immune cells that express the delta receptor are those immune cells that your body has specifically to cause immunosuppression when you need it. So if you if you have a chronic inflammatory state, you want to have cells that can balance that and create an anti-inflammatory state. When you're pregnant, right? So you have, in the setting of pregnancy, you will have these cells come into the placenta and essentially create an immunologic sanctuary for the fetus. The fetus has antigens that are made up from 50% from the father. And so it would appear foreign to the mother's immune system if the body didn't evolutionarily have a way to prevent essentially the mother's immune response from attacking the fetus. And so that was the big discovery. And the discovery is you have a single target that appears to control the ability of 3 different cell types that are important in the body for anti-inflammatory responses. So a single point of intervention that potentially can remove immunosuppression when you don't want it, like in the case of cancer. We'll talk about that in a minute. Or the contrary is, what if you need immunosuppression? You have Crohn's disease, or you have an autoimmune disease where you want to turn these cells on and make them immunosuppressive. Well, if you activate the delta opioid receptor, we know you can do that too. Our focus is predominantly in incorporating this into a strategy in the cancer immunotherapy space. So that's the major discovery. We're excited that the Scientific Selection Committee of the American Society of Hematology recognized the novelty of that discovery and provided us the opportunity to present it at an oral presentation. You know, they receive 8,000-9,000 abstracts every year. And in the area that we were focused in terms of the oral presentations, there were about 200 of them. And so, you know, that is pretty prestigious. It certainly demonstrates that the scientific community finds your findings of relevance to present them to the larger scientific academic community at large that attend these meetings.
So, Jim, the thing that I found most interesting about this program was that you're not targeting the tumor, but rather the tumor microenvironment and the you. All right, I'll just start that over. So the thing that interests me about this approach is that you're not targeting the tumor itself, but rather immune tolerance in the tumor microenvironment. Can you explain the mechanism of action of the delta opioid receptor and its inhibition, and how that fits together with that, and how it allows other drugs, you know, perhaps checkpoint inhibitors or other approaches to work to actually eradicate the cancer?
Sure. So you mentioned the tumor microenvironment, which is kind of a fancy way of saying where the tumor lives. So think of the placenta with the fetus, again, that model. So what happens when tumors form in your tissue, they secrete certain types of compounds called chemokines that attract anti-inflammatory cell types, MDSCs, regulatory T cells, macrophages, and these cells populate that tissue, and they secrete and or destroy a lot of the mechanism by which the body has an immune response to a tumor. And so it creates an immunologic sanctuary around the tumor. We are particularly focused on a cell type called MDSCs. They are the big culprit, right? These cells secrete multiple soluble factors that essentially inhibit the ability of your immune system to become activated, or if it is activated, it inactivates it. And they call that T cell exhaustion in one setting. And so we've demonstrated that a single point of intervention, so if we block the delta opioid receptor, and we'll start with MDSCs. On MDSCs, we shut off multiple ways for these cells that they create this immunologic sanctuary. If you can remove that shield that's around the tumor, then drugs like checkpoint inhibitors or T cell therapies, CAR T, TILs, they should all be able to work better. They should be able to continue working, not develop this exhaustion where they stop working. And I think that the reason that is so compelling is that a lot of companies are going after each of those soluble factors that these cells produce as individual targets. whereas we have a single control point, kind of a master switch. If we shut it off, the majority of them go off at a gene level and at what we call the protein level, the factors themselves. So, that's a very novel way to approach an area that a lot of pharmaceutical companies are trying to approach because it's kind of the holy grail, right? Checkpoint inhibitors stop working in that setting. T-cell therapy stop working. And it isn't related to the tumor. It's related to the tissue in which the tumor survives and thrives. And MDSCs are just one of the cell types. There's 2 or 3 others. We mentioned macrophages and we mentioned T cells. They all play a very important role in creating this sanctuary, this immunosuppressive environment for the tumor to grow.
So one of the reasons why there was so much interest in your delta opioid receptor work was because last year's Nobel Prize for Medicine was granted in this area based on work done on T regulatory cells. The 3 recipients had done work on how the immune system responded to Tregs, and then also the role of the FOXP3 gene. And I wanted to throw that in your court once again to explain the relationship between your work on the delta opioid receptor and the findings from the recipients of the Nobel Prize in Medicine last year.
Yeah, great question. We were excited about that because it essentially validates a lot of the work that we're proceeding with in terms of the targets like MDSCs and macrophages. So you may recall your immune system when you're born is called your innate immune system. That's your first line of defense, both when you're pregnant, i.e. when you have the placenta to create that immunosuppressive environment. And then the second line of defense is your adaptive immune system. And what the Nobel Prize was based on was that more advanced portion of your immune system, called your adaptive immune system, has these cell types, as you mentioned, called regulatory T cells. And they induce a sense of tolerance to potential normal tissue antigens. And the reason they do that is you don't want to have, for example, the importance in an autoimmune disease, a lot of times your immune system stops recognizing your normal tissue as being normal, as being self, and attacks it. And so regulatory T cells, when they activate the FOXP3 gene, FOX3P gene, I got to go back and make sure.
Yeah, I think it's FOXP3. I mean, I think I've said it both ways. Yeah, FOXP3. That's what it is. There's so many alphanumeric designations for genes.
All the mutations too. Okay. So when the FOXP3 gene is activated in these regulatory T cells, they become highly immunosuppressive and they induce this tolerance. And the reason that's important is if you can activate these regulatory T cells to be immunosuppressive, they can have great applications in chronic inflammatory disease, et cetera. So what's relevant to what we're doing with the delta opioid receptor is twofold. Regulatory T cells have a gene that produce opioids. It's called PENK3. And the PENK gene, by producing an opioid, must have a role in immunosuppression. And in fact, it does. The delta opioid receptor, when activated in regulatory T cells, will activate the FOXP3 gene and cause those cells to be immunosuppressive. When the delta opioid receptor is activated in macrophages or in MDSCs, which is our target of interest, it will cause those cells to become immunosuppressive. So now you've demonstrated that the delta opioid receptor in 3 different cell types that the body uses for anti-inflammation capacity or immunosuppressive capability is all linked together. So you have this, what we call convergence between your innate immune system, your adaptive immune system, and the ability to control immunosuppression if you want it, or inhibit immunosuppression if you want to. So Tregs, macrophages, and MDSCs are all very important in tumor biology because they create that immunologic sanctuary. We can intervene with a delta opioid antagonist. We can shut off all 3 of those cell types and remove that, what we call that immunosuppressive tone or phenotype in the microenvironment. And that's, for us, kind of validating work. Nobody's demonstrated this on MDSCs, macrophages, et cetera. And I think the linkage to the gene, the immunosuppressive gene, that was the basis for the Nobel Prize, really underscores kind of the potential of the technology that we're focusing on.
I think I was fascinated the most by the ability, it seems like, to tune the immune system. As you mentioned, you have autoimmune disease, or you could have, pathogens taking over, or cancer on the other extreme. And you can, by touching on the delta opioid receptor, even inhibiting it, or, somehow otherwise adjusting it, you can actually kind of find the balance in between those two extremes. Is that the right way of thinking about it, or is that too simplistic?
No, that's spot on. You've developed specific selective antagonists, you can shut it off. If you develop specific selective agonists, you can turn it on, and you can do exactly what you just said.
Okay.
It's a balance between anti-inflammation or immunosuppression and having a non-immunosuppressive environment, non-inflammatory environment like you see in cancer.
Yeah, it's an exciting time for the technology. So, well, that's great. I mean, I think that's fascinating how what you're working on tied in with last year's Nobel Prize in Medicine. So I wanted to touch on one of your other assets, which is your anti-VISTA asset, which you've designated now TBS 2025, I believe.
Right.
And so you acquired that last year from Kineta, and you've now folded it into the operations at TuHURA. What have you accomplished so far now that you've had it, and what are your plans as we look forward in 2026 on advancing that candidate?
Sure. You may recall a lot of my background really was in blood-related malignancies, so very familiar with VISTA and its role in what we call the myeloid compartment, those parts of your blood cells that essentially play a very big role in leukemia. And so we saw the potential for VISTA, given the research that was coming out in 23 and 24, that VISTA plays a very important role in why leukemias like acute myeloid leukemia, or AML, doesn't have an immune response against it. It's the primary reason that AML is not cured with chemotherapy, and why AML, when you relapse after a remission with therapy, essentially is resistant to subsequent therapies, and those patients unfortunately don't survive. And so if you can actually activate an immunologic response against leukemia, you may have a very potent therapeutic that can add to the current armamentarium that physicians are using to try to cure AML. The only time you see an immunologic response, in a very potent immunologic response, is in the setting of an allogeneic hematopoietic stem cell transplant, because you have a foreign immune system now recognizing the leukemia, what we call graft versus leukemia effect, which is very potent in eradicating leukemia. But you don't see that in a non-transplant setting. And so that's really been kind of our interest. We think, VISTA is there for a reason biologically, normally. It is the only way that you keep your T cells quiet. I mean, that is its primary role. And like everything in cancer, it looks like leukemic cells have hijacked that to keep your T cells quiet, so that those cancers can survive without having an immune response against them. So that's why we were interested. That's why we went after that asset pretty aggressively. I think we acquired it at wholesale valuations, if you will, and we are in the process of putting that into a phase 2 study in AML.
So the initial clinical work that was done on the anti-VISTA asset was actually in solid tumors, and you're working in blood cancers with it. What are some of the reasons that you think it's more beneficial to use it in the blood cancers rather than the solid tumors? And does it have a potential at some point in solid tumors, or do you think it's so beneficial in the blood cancers that's going to be the direction that it'll remain in?
I mean, good question. So again, I've grown up with the philosophy, in at least when we were involved in pioneering the whole transplantation space, that things are there biologically for a purpose. And VISTA has a purpose. And what I mentioned was the myeloid compartment and on keeping T cells quiet. That biology, pharmacology, molecular biology associated with it is pretty well described. in solid tumors, it's fairly amorphous. They don't know if VISTA is playing the role as a ligand, is it playing the role as a receptor, is it activating PD-1, is it inhibiting PD-1? So it's pretty complicated, as you might imagine. Solid tumors are very complicated, right? Multiple mutations, different types of subtypes of cancer, lung cancer, for example. And so, my bet would have been that you're not really going to be in a position to know which tumors and how best to apply a VISTA inhibiting antibody in that setting. Because again, the literature, while it's now evolving, is still very confusing. And so that bet happens to be correct. And right, there are 4 VISTA inhibiting antibodies that are in development. I mean, Hummingbird has had one. They outlicensed it. Sensei had one. Kineta had one. And then there's another company.
PharmaCine, I think, maybe?
PharmaCine, right, they have one. And they all went into solid tumors. Every single one of them did exactly the same phase 1 trial, right? And they looked at safety, and they looked at it in combination with pembrolizumab or Keytruda, which is its brand name. And they all show the same thing. You don't get a response. It's safe. As a matter of fact, the Hummingbird antibody, they gave it at 1,400 milligrams weekly. I think whatever you have to give 1.5 grams of anything, it can be safe, but it has to show a signal in efficacy. Seeing one response out of 44 patients is, in my view, if you had a go, no-go decision, that would be a no-go decision, right? And so that's why I think, quite frankly, it's a very important therapeutic potentially, but I think its relevance for its application is going to be in hematologic malignancies, not in solid tumors, where the biology is much more defined. For example, just last year, there's a subset of AML that are driven by a number of what they call gene rearrangements or mutations. And NPM1 happens to be a very important mutation in AML, and about 1/3 of them, and it drives the leukemic signal. Now, there are small molecule drugs that have been approved that inhibit that signal and actually have shown benefit in a relapse setting in about 20% of patients, but they still have very short duration of response and relatively low complete response rate. And the primary culprit seems to be VISTA expression on the leukemic cells. And that data was shown that if you now take VISTA out of those cells, that those leukemic, the leukemia in your animal models, now you have pronounced survival advantages and you show you activate an immune response. So it's biologically reasonable and sound to look at what's the application of VISTA in NPM1 mutated AML who are getting a menin inhibitor. Those are the small molecules I was mentioning. Can you improve the response rate and duration of response if VISTA truly is playing a role on immune evasion for leukemia, for AML? And that's a quick answer. You don't need 100 patients, 14 patients, you'll get a go, no-go decision. And that's what we plan on doing this quarter, starting that trial.
Okay. So TuHURA has an antibody-drug conjugate construct. And does it make sense to combine your anti-VISTA TBS 2025 with your delta opioid receptor to use those in combination?
Yep, and absolutely. So I mentioned that VISTA is overexpressed on the myeloid compartment. That includes expression on MDSCs, macrophages, and in the setting for AML in leukemia. And so if you're able to shut off immunosuppression by blocking the delta opioid receptor and then blocking VISTA on, for example, B cells and T cells, you now essentially allow T cells to wake up. They're no longer sleeping or quiescent from the role of VISTA. And that should give you a very potent ability to remove immunosuppression and activate an immune response. And we think that that's a totally novel. Matter of fact, we know it's a totally novel approach. We are the only company, to my knowledge, that are developing immune targeting antibody drug conjugates instead of tumor targeting antibody drug conjugates. And I can go into that in more detail if you like.
Yeah, that sounds interesting. Well, I was going to also mention that ADCs are one of the most attractive areas in oncology, and there have been a number of acquisitions there. And in fact, I think last November, Mersana Therapeutics, they got bought out or acquired for about $285 million. And I'm wondering what does that say to you about where to focus your efforts and what the market sees as having value, especially in the ADC space.
Sure. I think if you turn the clock back, you look at the Pfizer acquisition of Seagen, right? Seagen pioneered this whole field after Pfizer bought Seagen for $43 billion.
Right.
Amgen bought a single asset ADC from a Japanese company, I'm blocking out the name, for $3 billion. There were a host of acquisitions in the multi-billion-dollar range, including one for $1 billion in a preclinical stage company. The novelty of what we're doing, and this is why I think this will come under a lot of positive scrutiny, especially now with the scientific validation at ASH from the pharma community. And I'll say why. The original application for an antibody drug conjugate, so they use an antibody to target a target on a tumor cell, and they carry with it a payload. The first generation had payloads which were cell toxins, right? They would kill the cell. And the way those ADCs would work is that the antibody gets taken into the cell, right? So it has to be internalized, and it carries with it the payload, which is the chemotherapy, if you will. So that was the first generation, which used an antibody cytotoxic conjugate. Conjugate just means combining it to, or linking it to. The next generation looked at, well, if chemotherapy kills tumor cells, there are other ways to do that. There are protein degraders. There are ways to inhibit the cell cycle, i.e. prevent these cells from dividing. That was kind of the next generation, right? The non-cytotoxic, but they still target a tumor, get internalized, and then prevent the cells from dividing. The last generation and most recent were called checkpoint ADCs. So they'll take VEGF, which is a target on lung cancer, for example, and they'll tie to it Keytruda, PD-1, an anti-PD-1 checkpoint inhibitor. And again, that gets internalized. The PD-1 essentially blocks the ability of the tumor to prevent a T cell from being activated. Those are called checkpoint ADCs. You notice they all have one thing in common. They all target a tumor. We have what we call immune modulating ADCs. So two differences. First thing is the antibody that we're using, VISTA, that's targeting the VISTA receptor on T cells to allow them to wake up. And we're linking that to the drug. And in this case, the payload isn't a cytotoxic and it isn't targeting a tumor. It is actually targeting the delta opioid receptor. So we have the first set of drugs that antibody drug conjugates that are not internalized. They are not cytotoxic, but in fact, they will target the microenvironment. They'll bind to this delta opioid receptor on immune suppressing cells, remove that immunosuppression, while at the same time VISTA releasing T cells from being kept quiet and allowing them to become activated. So our ADCs target the tumor microenvironment, not the tumor. And we're the only ones, to my knowledge, that are doing that. I certainly know we're the only ones doing it by targeting the delta opioid receptor linked to a VISTA inhibiting antibody. That I'm confident on, because if you look at any of the patent literature that's out there, it's all us.
Okay, yeah, that's fascinating. Again, we're going back to that targeting the tumor microenvironment to allow the immune system or other drugs to actually fight the tumor rather than going at it directly with your candidates. So I find that fascinating. As we just mentioned, there's M&A going on. It's been pretty. It's increased substantially in the second half of last year. I think I have some stats here. There were, according to my numbers that I looked at this morning, and Evaluate Pharma, which is they pulled together a lot of data in the industry, but they found $165 billion of transactions in 2025 compared to about $118 billion in 2024. So, there's a nice increase year over year, and we've seen it with some of the things that we already discussed. And, I think a few more, let me just mention, there was Blueprint Medicines, and Springwork Therapeutics, in the multi-billion-dollar range. And then there was also Mersana Therapeutics and ADCs. What do these transactions suggest to you about what's going on and what we could see in 2026? And also I want to, mention that the XBI, which is one of the biotech indices that's out there, that's done pretty well so far. Last spring it was in the 60s and now it's in the 120s. So There seem to be some green shoots perhaps out there. What does this make you think about 2026 and the environment for, small life sciences companies?
Sure. So I'm going to start that on two fronts. I'm going to start that on relative comps. So if you look at companies that are, for example, I mentioned Replimune, right? So they're targeting similar technology to IFX, distinctly different, but still going after essentially the same concept of making tumors sensitive to your immune system and allowing checkpoint inhibitors to work. They have a market cap in the BLA filing now. They have 6 months before they have their so-called PDUFA date, about 850 million. If you look at them when they entered phase 3, their market cap was around 350 million. If you look at companies that have essentially produced, developed, and then commercialized these menin inhibitors, right? You mentioned it, AML, relapse refractory AML, and they started in the same thing. When they first put their drugs into phase 2, their market cap was in that $350 to $450 million range. They're now in that $1 billion and a half for the setting for Syndex. We're focused in those same two areas. So from a valued gap perspective, we are very undervalued in my view. Clearly, on the comp side of it, we should be somewhere around 3-4x on the low end, maybe 6-7x on the more median, kind of where market cap should be for phase 3 companies that are targeting the targets that we're targeting, and then certainly the AML side of it. So I think that's the first potential in terms of kind of this asymmetric return, if you will, when the market starts to recognize comms and what we're doing. Now, that with regards to your question about M&A activity, you have companies like J&J that are developing menin inhibitors that have made a real push in developing, moving into the hematologic franchise, if you will, hematologic malignancy franchise. They now kind of own that space in multiple myeloma. They're clearly moving forward in the AML space with Bluxamenib, which is their very novel menin inhibitor. It seems to have the highest response rate associated with it. So if a VISTA inhibiting antibody, being novel, being the only one being developed in AML, can improve response rate and duration of response by being utilized, like all chemotherapy and treatments always end up being in combination, I think that would be very synergistic with the portfolio direction that a company like J&J is doing. Even companies like Syndex, quite frankly. And here's the rationale. A menin inhibitor gives you a 20% CR and it lasts for 4 months on average. Then they come off your menin inhibitor. Well, the VISTA and a menin inhibitor combination gives you a 35% CR rate and they stay on it for a year. You can do the math, right? That's not even organic growth. That's growth by the basis of the combination of the two drugs. So we think that VISTA will become very attractive in the hematologic malignancy space to a number of big pharma companies. Let's take a look at the other side of the equation. If you look at Bristol's website, AstraZeneca's website, Lilly's website, and their oncology program, they're all focused in the microenvironment. MDSCs are a really hot target. And if you take a look at their programs, matter of fact, one of the leaders, we'll call him Mr. MDSC, runs immuno-oncology worldwide for AstraZeneca. This is Dmitry Gavrilovich, who worked here at the Moffitt with Alan List on defining this group of cells. We're the first to define a new target on MDSCs, a new target that may play a very big role in the microenvironment and for intervention. So I wouldn't be surprised if the data that we are generating on inhibiting the delta receptor and the potential to link it to a VISTA inhibiting antibody will have a lot of pharma traction as we start to show proof of concept moving forward. So, I think you're going to see a lot more M&A. I think rather than worried about loss of exclusivity, like for example with Keytruda, like Amgen did so successfully with Neupogen making Neulasta, there's ways to maintain that franchise despite the potential for generic erosion. I think you're going to see more of the vertical growth than the horizontal growth, where they're going to want to take an existing franchise and essentially expand it. So if I'm a therapeutic area focus of AML and I have a menin inhibitor, what else can I do with it? how else can I own more of that AML space and make major advances and get further approvals further up in terms of frontline as opposed to relapse refractory. So I think we are primed for that being a potential exit strategy, even though it may not be what we're looking at today, but certainly not too far in the future, as both of those programs, the VISTA AML program and then the proof of concept with ADC, start to attract attention. Next year, it's all IFX in the phase 3 and those results. And then I think very much like Replimune in terms of shareholder reward when their data was positive, and the subsequent, what will be the FDA approval, you see significant value creation along with drug approvals in this industry. And so I think you'll see a lot more M&A activity. A good example is what was it called? Dark Blue, or the one, Dark Blue,
Right.
acquisition.
Yeah, which extends that theme of AML and M&A. I think that was just announced recently. So what do you think of that? I think that's in protein degraders. They're working on AML. The amount was, actually I wrote it down somewhere here, some $840 million with total value. I don't think they broke it down between upfronts and milestones, but what does that say to you? And how do they fit into the whole AML situation?
Yeah, I mean, it's spot on with what I was saying, right? That's a preclinical compound, hasn't even been proven in the clinic. It's another potential mechanism for interfering with the leukemic signal that drives AML. And so you're going to see more and more pharma companies that are focusing in the blood-related cancer space. We used to call it low-hanging fruit because there's so much of an unmet medical need. And it's, unfortunately, it's a high incidence disease. And you may say, well, 15,000, but it's fatal. If you're not eligible for a transplant, all they're going to do is give you some time without your disease, i.e., progression-free survival, but not overall survival. And so I think you see a lot more focus in that blood-related cancer space because molecularly you can define a better way to interfere than you can, for example, in solid tumor types like lung cancer. If it wasn't for checkpoint inhibitors, there really hasn't been a lot of advances in solid tumors. Maybe ovarian cancer, right, with all of the PARP inhibitors and some of the other targeted therapies that they have. So I think, we are right in the sweet spot where pharma will be looking for novel approaches to blood-related cancers like AML.
Okay. And, kind of extending the discussion of markets improving and M&A, one may think that perhaps the financing environment would improve too. And, on that same topic, TuHURA recently closed part of a financing, and you structured it in a little bit different way than I think some other companies have. Why don't you tell us about that? And also your thoughts on the financing environment as we move into the new year.
Yeah, so let me start by saying we've been fortunate as a company to have a very supportive and kind of believing in the technology, believing in management investors, right? And we are almost exclusively, I'd say 40-50% of the company is supported by ultra high net worth individuals that have really financed this company going forward, both in the past and presently. So we wanted to start to expand out into kind of the institutional base. We had one institution that approached us that would anchor a transaction. And we went forward with that institution and then had a handful of our existing investors come in around that. And so the 15.6 million, 7 million of that, one investor said, kind of coming into the end of the year, what if I give you 5 million at the end of January and 7 million and 2 million in February? And we're like, sure, you want to defer it for essentially 45 days or 2 months. That's not an issue for us, wasn't an issue from the security side of it, and certainly wasn't an issue from the other fund that was participating in the round. But that, again, that was supported by our shareholders. We had one shareholder we took a little bridge loan from just to get us to this financing after the government shutdown, because we would have done this 2 months before. And he even said, look, I don't want the money back. I'll just take it as equity because I kind of believe in the long-term vision for the company. Unfortunately, the market took that the wrong way. We got kind of penalized, not for fundamental issues, right? We're firing on all cylinders, as some people would say, hitting our milestones, advancing the programs. We shouldn't be down here in both the price and market valuation range, and I think that will change as again the market and quite frankly the buy side institutional environment starts to recognize the value play here in the company. I think, like you said, you know, the biotech index from April to the end of the year outperforming the S&P, outperforming NASDAQ. I think that's a very bullish signal coming into 26. I think overall for the year, it's probably the first time in 3 years where it hasn't been below the major indices. So I think we'll see more of that coming into 2026 following, you know, we're at JP Morgan next week. Good example, good barometer. We have 36 meetings in the 36 meetings in the 2.5 days that we're there, with more than half of those being, you know, brand name buy side funds.
Well, that's impressive. Yeah, I guess, you know, next week is the big week in biotech in San Francisco, and with that, you know, I think we could move on to some kind of broader themes in the industry. You know, one thing that's been coming up a lot is China's surge in the life sciences. You know, they have a ton of trials going on, and some are concerned that they may take the mantle away from the United States as being the leader in the life sciences and biotech space. And I wanted to see if you had any thoughts on that, and you know what perhaps the United States could do to maintain our lead and perhaps even improve on that. What do you think about that?
Right. So, you know, I have a reputation of being very transaction oriented. Right. We have developed, with the exception of the delta opioid receptor technology, we have not had any of the drug candidates that I have brought in-house that have been subsequently approved and to have become kind of the standard of care in blood-related cancers. And so I get a lot of incoming, at least once a week, from a variety of banks, Chinese banks that are brokering Chinese companies for business development, looking at some very interesting assets, some of them later stage, meaning phase 1, some of them still being IND-enabling studies, et cetera. It's very exciting. It's very interesting, I think, but it's also there is some risk associated with it. If you look at clinical trials that are done exclusively in China, there are differences both genetically in terms of how populations metabolize drug. There are differences in how clinical trials are conducted at clinical sites, and so some of that data I am confident. While it may be exciting to bring it in-house and say, well, we have proof of concept, for example, in a human lung cancer study with 90% response rates, et cetera, that may not be translated into the heterogeneity of the American population, the U.S. population. And so I think the jury's out on essentially the utility of those novel drugs in that have shown clinical data, whether or not that translates into U.S. data. With regards to other assets that are available, it's quite frankly, I don't want to say no different, but you find them not just in China coming to the U.S. Quite frankly, there's a number of European companies that are purely on the development stage that are licensing products out, you know, and then we will see. Some of them are very intriguing, but they're not alone. You know, for example, I got approached by a CCR8 / bispecific targeting regulatory T cells in the microenvironment. And if you go and you just look in the literature, there's probably half a dozen of those. Why you would select one from a Chinese company if it's more advanced, the data, et cetera, relatively inexpensive. In my view, anything that advances the field forward and with good science and good therapies, doesn't matter to me where they come from. I don't see this, quote unquote, as a threat to the U.S. Innovation. In fact, I'd probably see it as being complementary to what biotech and what the pharma companies are doing with their own portfolios. I don't think it's going to be either or. I don't think they're mutually exclusive.
Okay. Thank you for your view on that. And another emerging topic that's out there is the most favored nation pricing. I know it's a little bit early for TuHURA to be thinking about pricing, but you are an executive in a company with three assets that could be on sale in a few years. And just to let our viewers know, the most favored nation pricing would take prices in other developed countries and then apply them to those prices paid in the United States. And as probably most people know, the prices we pay for drugs here is higher than elsewhere. So this creates a risk for launching new products outside the United States. And I'd love to hear your thoughts on how this might affect a company and the way they commercialize their product, both in the United States and outside the United States.
And I've had some experience, as you may recall, with at least one of our drug candidates. I was the person who did the defense at NICE, which is the organization in the UK that determines reimbursement and pricing, as well with GBA in Germany, et cetera. So I think where the whole field is moving or trying to move is in the whole health economic outcome perspective, right? They used to call quality adjusted life years. So what value does your drug add? And the value is now being equated with regards to outcomes. So if you have a response rate, and there's 9 other drugs that have a response rate, but the response is of short duration, the drug may be approved, but what's the ultimate benefit if it's a surrogate endpoint? Doesn't mean it won't get reimbursed. The question is at what prices get reimbursed. If you are truly adding and driving value to a patient outcome, and I know this, probably the most challenging environment has been the UK for drug reimbursement. And I think they are also kind of the reference for a good portion of the European Union. In terms of other countries, will just draft off of their opinions in terms of pricing. So I think that's going to become probably a more important aspect of your drug development plan when you look at a target product profile for a compound. Me Too's are not going to be competitively priced in the United States because they won't be competitively priced in Europe. Particularly the UK is probably going to be the common denominator. And so I think that's really what's driving the development in terms of endpoints, et cetera. So for example, in Merkel cell, if you have 50% more of those patients respond, that's good. Is that going to yield a premium price overseas versus here in the US? Maybe not. If that translates into an improvement in an endpoint that is clinically meaningful, like progression-free survival, then it will plug into their formula the way you would want it to, that would at least justify the right pricing, i.e. the premium pricing that that product would deserve in Europe and here in the US. So I think that's the direction that pharma companies are starting to look. I wouldn't say that that's exclusive to this administration. I think that has been happening for the last 10 years as companies have been working in health economics into their plans in general, in advance of essentially their phase 3 program. So they have an understanding of the arguments that they need to make with the healthcare reimbursement communities with regards to what their products add in terms of value for patients and their outcomes.
Okay, and I thought I'd wrap up with another one of the leading themes in the life sciences space, and that's the patent cliff that's coming up. There are a number of very popular products that are rolling off patent. Kadcyla is one, and then of course Pembro is going up. Keytruda is coming off patent as well, and these companies need to replace their pipeline with new products. So how does that also inform what your thoughts are on 2026 and kind of the activities that'll take place?
A lot of people thought there'd be a fair amount of acquisition from Merck looking at trying to potentially fill that earnings gap, right? Because 40% of their earnings is Keytruda driven. And like Bristol did with Opdivo, Merck did the same, developing a single injection, long acting versions, kind of like the GLP-1, Novo story, take a pill instead of injection. And I think there's good examples in the industry of how that has been very successful in maintaining 80% of the franchise as opposed to the more traditional 50-60% generic erosion. A good example, I mentioned, is Neupogen. Neupogen is given frequently, typically was given in the hospital. Then they developed a subcutaneous injection, but still frequently. And then they came up with Neulasta, where you can give it once a month or once every 2 weeks from home. And that prevented the erosion, generic erosion of essentially that neutrophil growth factor that Amgen had, which was a big portion of their franchise back then. Same thing with erythropoietin, right? So EPO, and then subsequently their long-lasting pegylated version. So I think we're seeing that, or does that advance the field forward? No, I think that is a mechanism by which a pharma company is going to try to retain market share by just changing the form. Is it more patient-friendly? Sure. Does it work differently? No. Is that advanced for the patients and their outcome? No. Will they be able to maintain a premium pricing on that over generic? Definitely, because they'll have exclusivity for that use, and it will be adopted by the treating community because, again, it's more friendly to them. And quite frankly, the payers like it better because you don't have the clinic visit for infusion and for physician charge and nursing administration charge, et cetera. So I'm not as focused on that cliff. Certainly a lot of the analysts, when they run their forecasts in terms of market share erosion for Keytruda in particular and how that may hit Merck's EPS and ultimately, obviously, their valuation. These companies have a lot of people that focus on that way in advance. I think you'll see that, Pembro sales will come down, but it's not going to come down 50 or 60% like it used to be. And so I don't see as much, how do you put it, concern about that in the sector anymore as these other drugs, these long-acting versions of them become available or alternative formulations become available. That adoption rate is going to be higher than essentially adopting generic versions of it.
Okay. Jim, well, thank you for joining me today. I enjoyed speaking with you. And for our viewers, we had the CEO of TuHURA Biosciences, Dr. James Bianco, ticker symbol HURA.